Okay, students, we're going to take a look at glycolysis today, and we're going to look at the 10 steps of glycolysis. Remember, glycolysis is the first part of aerobic cellular respiration in which glucose is partially oxidized, and we make a little bit of ATP and a few other things along the way. So what I'm going to show you are the first few steps of glycolysis, and then we're going to walk through all 10 steps in a series of short videos. So this is glycolysis, and remember that glycolysis is happening, all of the steps are happening outside of the mitochondrion in the cytoplasm. So all of these molecules and enzymes are in fact in the cytoplasm. All right, so let's take a look. Remember, there are 10 steps, and we start with a nutrient. We're going to start with glucose. This is the linear form of glucose. Glucose's molecular formula is C6H12O6, and its linear form, it looks like this. There's a 6-membered ring, and one of the carbons is sticking out of that ring. Notice that there are a lot of hydroxyl groups. Remember that from chapter 5. And there's our nutrient. That's the food that we've eaten, the Pop-Tart you had for breakfast, right? Okay, so what happens during glycolysis? The first thing that happens is, let's write down this as step 1. In step 1, glucose gets converted into glucose 6-phosphate. Okay, what do you think that 6 means? Well, it means that a phosphate group is added to the 6th carbon of glucose. And you can see it up there. There it is. There's our way to write phosphates, a P with a circle, right? So in this step, we add a phosphate to glucose. This is going to require an enzyme. There are enzymes for every step in the process. The enzyme is called hexokinase. And by the way, we're on PowerPoint slide 11, if you're looking at your PowerPoint slide. I'm just going to write down that it's a kinase enzyme, and the enzyme goes above the arrow typically in a reaction, right? And so where did this phosphate come from? It came from, this seems strange, people, ATP. ATP donates a phosphate to glucose. It loses that phosphate and then becomes ADP. ADP is adenosine diphosphate. It has lost a phosphate. It gave it to glucose. Uh-oh. We're supposed to be making ATP. Yeah, we're not, right? Looks like we're losing or investing in ATP. This is an energy investment step. So I'm going to write down a couple of things about this step 1. The first thing we did was we invested energy. Energy invested, 1 ATP. So you can put in your notes, used 1 ATP. Second thing is we phosphorylated glucose. We added a phosphate to glucose. Not sure why, but maybe it's important. So we phosphorylated glucose. And number 3 that you want to write down is we used an enzyme called a kinase enzyme. And in your notes somewhere, we want to remember that name. That kinase enzyme is an enzyme that is involved in the transfer of a phosphate from one molecule to another. So the kinase enzyme is involved in the transfer, helps out, catalyzes the reaction, catalyzes the transfer of a phosphate from one molecule to another. And whenever we see the transfer of a phosphate in a reaction, we know that there's going to be some type of kinase enzyme involved. Okay, so step 1, we've lost some ATP, but we've added a phosphate to our molecule. What happens in step 2? Step 2. In step 2, glucose 6-phosphate gets rearranged into fructose 6-phosphate. This is simply a rearrangement step. Fructose is a 5-membered ring, and so you can see 2 of the carbons are outside of that ring. There's our phosphate, all right? This step I think of as a rearrangement step. Glucose 6-phosphate is simply rearranged into an isomer. Oh, in fact, the type of enzyme that's used here is an isomerase. It's called phosphoglucoisomerase. I'm just going to put a few dots there and then write isomerase. You don't have to memorize all of the enzyme names, but you do have to know several types of enzymes. You should be able to recognize when a kinase enzyme is used, for example. Okay, now we've rearranged this molecule in step 2. What's going to happen to fructose 6-phosphate next? Step 3. Okay, in step 3, fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. Oh my. Fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. What do you think the bis means? Two phosphates. And fructose picks up a phosphate. Oh, here it is. So now it has 2 phosphates on it. Where do you think it got the phosphate from? Well, this is another energy investment step. In this case, ATP donates a phosphate, just like step 1. ATP donates the phosphate to the fructose molecule, and now we have a fructose molecule that has 2 phosphates attached. What kind of enzyme do you think is used here? Phosphofructokinase, a kinase enzyme. All right, so let's write, because kinase enzymes catalyze the transfer of a phosphate from one molecule to another. All right, let's write down a few things about this step because it is kind of important. We did some energy investment. So another ATP has been used, 1 ATP used. Okay, 2, we phosphorylated. That's such a weird word, huh? Phosphorylated fructose again. And we used a kinase enzyme. Okay, what's going to happen to fructose 1,6-bisphosphate? We'll find out in the next video.